And I'm just getting us set up recording. Okay, there we are. So today we're going to finish up with our little piece on mean arterial pressure on MAP and its regulation. Then we're going to move into talking about the blood. So we're moving into chapter 15, I believe it is today. So for our warm-up question, what we're trying to do is predict the effect on MAP if our sympathetic activity decreases. Okay. This is basically kind of the reverse of that flow chart we traced at the end of Monday. So we know that MAP is dependent on our heart rate, our stroke volume, and on TPR. So we're trying to think what effect sympathetic activity has on any or several potentially of those variables. So one of the ways we can think about this is by looking at that handy dandy flow chart. So this is the reverse of the situation that we are given in this question. Pull up my drawing tool. Okay, so here we can see that this chart is for increases. Why is this not working? Okay. Hearing some people aren't seeing the slides. I am going to reshare that. Try again. Sorry about that. Oh no, guys. Okay, that means you didn't see this question either. I apologize. Okay. Well, can you see them now? Can people let me know? Okay, awesome. Okay. In that case, I'll give you a second to write down your answer of what you think is going to happen to MAP if our sympathetic activity decreases, because I think I didn't quite give it away yet. Okay, so you got a quick predicted answer, at least maybe in your mind, right? Okay, so continuing with my explanation. So we were looking at this flow chart, right? And we can kind of run it in reverse. So in our flow chart here, right, we had our sympathetic activity going up. What we're thinking about now is like, okay, so what would happen if instead it went down? So if we had this sympathetic activity go down, right, so instead it goes down, fundamentally, we're looking for what the effect on MAP is. So at the bottom, you can see that when sympathetic activity went up, MAP went up. So immediately you could just skip all this business in the middle and say like, well, okay, it's going to go down, right? If I flipped one arrow, I'm going to flip all the other arrows. So that's true. So that would be a shortcut from just looking at this picture. But I do want us to kind of understand why that is the case. Okay. So if our sympathetic activity goes down, that means all these little effects in the middle, right, between our sympathetic activity and MAP are also going to be affected. So instead of raising the heart rate by increasing activity of the SA node, as we ease off the sympathetic activity, right? So basically when we say if sympathetic activity goes down, we're talking about relaxing there, right? Sympathetic activity is fight or flight. So if you're relaxing, maybe you're trying to go to bed, or you're just coming down after a high anxiety period, right? Your heart rate's going to go down as one of the effects of sympathetic activity going down. So our heart rate going down. Remember, right, our equation, I better type this, right? Heart rate by stroke volume by TPR. So those are the three things we're thinking about when we're thinking about things that can affect MAP. Okay, so as we're relaxing, our heart is beating slower. So that's one way that MAP's going down. We're also affecting stroke volume by affecting contractility of the heart, of the ventricles, and a little bit by affecting the venomotor tone. So basically, our heart isn't going to contract as hard when we're not in these situations of high stress with this high sympathetic activity. So our we're working the heart, right? We still need blood to flow around, but not quite as urgent, not quite as much, not quite as fast. So that's bringing stroke volume down. And because of some effects on the veins, right, we're letting veins go back to their normal high compliance state. So we're not putting this pressure in the veins that causes the heart to fill more. So that's another way we're affecting stroke volume here. So that's also going down. Right, and you can see every time, whenever you're looking at one of these diagrams, right, you can just go through and reverse all the arrows to see the opposite effects. Right. This is a lot of arrows to draw in the central one, but we could do it. Okay. And finally, we are also affecting TPR. So we've decreased heart rate, we've decreased stroke volume. Conveniently, we're also decreasing TPR. Right? Remember sympathetic activity, giving off norepinephrine, binding to alpha receptors, causing vasoconstriction of the arterioles, therefore increasing TPR. For easing off, we don't have that norepinephrine floating around anymore. We're not going to have the same state of vasoconstriction. So that's the way we should be thinking about questions when we're thinking about what affects MAP. I want you to have this understanding in your head. You don't have to have this exact flow chart memorized, but we're just trying to get to how any changes affect these three factors at the bottom: heart rate, stroke volume, and TPR. So that brings us to our regulation of MAP through hormonal control. So what we talked about on Monday was basically neural control and this baroreceptor reflex, right? That was actually what this original drawing was for. This reflex where baroreceptors in your arteries sense a sudden drop in blood pressure. And so they immediately pump it back up a little bit because that's dangerous. You don't want to faint. Okay. But we can also have more long-term regulation. So this isn't the same as the baroreceptor reflex. This is going to happen more on the order of longer term, days, weeks, potentially. So this is where we're going to have some of the renal regulation of blood volume come into play. We're not going to talk about what the kidneys are actually doing yet. That's the renal part. But these hormones we're talking about are involved in the kidneys, at least two of them. Okay. So hormonal control is going to involve epinephrine, vasopressin, and angiotensin 2. Okay. Epinephrine is still kind of acting on the short term, right? We know that epinephrine released by the adrenal medulla, right, in response to sympathetic activity. So those things are very linked in our minds, hopefully. Sympathetic activity and epinephrine and the adrenal glands, right? So epinephrine is basically going to have the same effects that we saw when we were just tracing the sympathetics. So if we increase epinephrine, we're going to be increasing mean arterial pressure. So increases in epinephrine are basically bringing our blood pressure up, because epinephrine acts on smooth muscle in the arterioles, right? Just like norepinephrine, if we have enough epinephrine, we can bind those alpha receptors, causing vasoconstriction in most parts of our bodies. It is true that we want to remember that epinephrine causes vasodilation in like skeletal and cardiac muscle, right? That was kind of an aside. We talked about the alpha receptors versus the beta 2 receptors when we were talking about the function of the arterioles. But our overall big picture effect, if we're looking at the body as a whole, is going to be an increase in TPR, right? You don't want your total blood pressure to suddenly drop as you're trying to run away from an attacker or something, right? So the overall effect here is going to be an increase in TPR. And we're remembering our equation, right? Caps lock, heart rate, by stroke volume, by TPR. Tiny, that's fine. Okay. So we're increasing TPR, which would have an increasing effect on MAP. So we'd bring up pressure in that way. Also acts on the smooth muscle of the veins. So it increases venomotor tone, just like we saw norepinephrine from the sympathetic nervous system doing. So as we increase venomotor tone, we're decreasing their compliance, meaning pressure can build up in the veins, which makes the heart fill more, because the way the heart fills is a pressure difference between the veins and the heart. So that's affecting our stroke volume. And it also acts on the heart directly, epinephrine does. It increases the heart rate and the stroke volume directly by binding to receptors in the heart. So overall, epinephrine is a hormone, but has pretty much the same effects as the sympathetic nervous system does when we're thinking about its effects on blood pressure. So that shouldn't come as a surprise. Our new hormones that we haven't really talked about are vasopressin and angiotensin II. So both of these hormones are vasoconstrictors, and they're both involved basically with water balance, which we'll get into on our unit, on our renal unit in the kidneys. Fundamentally, the way that they affect mean arterial pressure in terms of their vasoconstriction is that, like other hormones, nerves that create vasoconstriction, they increase TPR. So that's one of their effects, but they actually have many effects, which aren't listed here. So you don't have to worry about them for right now, but they do have other ways that they also affect MAP, bringing it up. So basically they reduce kind of urine output by keeping up blood volume due to the way they function in the kidneys. Angiotensin II is actually pretty cool. We'll look at it in great detail, and I think it's our third unit. It also stimulates thirst, so it also stimulates you to drink more water. So these two will come back to, but they are working on the blood volume side as well as working on these sort of more short-term things. Okay. So the baroreceptor reflex we were talking about on Monday had to do with our pressure receptors in the aorta and in the carotid sinus, so in our arteries, right? We do also have receptors that are related to pressure, or related to blood pressure, I should say, in our veins. And we think of them more as volume receptors than pressure receptors because our veins are focused on being a volume reservoir. So we have cardiac and venous baroreceptors that we can think of as like low pressure receptors. They're low pressure baroreceptors, they're reacting to volume. So the places we'll see those are in walls of large systemic veins and in the walls of the atria, so where blood is filling into the heart. And they get triggered by a decrease in blood volume, right? So if you're losing blood or if you don't have enough blood volume, right? Blood volume is part of what's pushing on the walls of things to create pressure, right? So that can also be an indication of a drop in overall blood pressure if you don't physically have enough blood in those veins. So while they don't directly cause the baroreceptor reflex, the responses they trigger are going to be pretty much the same. We're going to have sort of synergistic effects reacting in parallel, the baroreceptor reflex to bring blood pressure back up. So when these get triggered, try to say it simply in a linear form because I always like having kind of steps, right? So if we have a decrease in blood volume, right? So we have small amounts of blood, our receptors get activated, which means that we're going to trigger responses that are going to bring MAP back up. So it would look pretty similar to that flow chart we had at the beginning, slightly different, but we'd expect to see some of these same effects essentially. Okay, so I said that for time purposes, we're going to go kind of fast through this, so don't expect to see these things on the exam, but they are neat and relevant to like patient care. So I do want to talk about them for a sec, but since you're here, make a note in that. I'm not going to focus on this stuff for the exam. But basically, there are other cool cardiovascular regulatory processes that exist and that are kind of related to blood pressure and respiration, chemical concentrations, and heat. Okay. One neat one is that there's this thing called respiratory sinus arrhythmia, which is that your heart rate actually changes as you breathe in and breathe out due to increase in sympathetic activity as you breathe in and increase in parasympathetic activity, aka relaxation, when you breathe out. So this means that you would see changes in your blood pressure as you're breathing in and breathing out, which is why you want people sitting calmly, breathing evenly when you're taking people's blood pressure, right? You don't want wild changes. And I hear target shooters actually use this for sighting better, like to make their shots as they breathe out, because then you don't have as many heartbeats, so you don't have anything like basically shaking you if you can feel your heartbeat moving through your arteries. So that's just a cool thing that exists that does have effect on heart rate. Okay, chemoreceptor reflexes is also kind of linked in with your respiratory system, but in a different way than with the respiratory sinus arrhythmia. So again, I hate saying this so much this lecture, but we'll talk in detail about the respiratory system in our next unit. But basically there are sensors that sense whether you have increasing levels of carbon dioxide in your blood, because that's bad because you want to get rid of the carbon dioxide. So your body has a bunch of mechanisms it uses to regulate blood levels of carbon dioxide that do affect your cardiovascular system and your blood pressure. Basically, your body is trying to increase carbon dioxide elimination, so to get rid of carbon dioxide if you have too much. And we'll talk about why that's really important as we move forward. But some of the things it does in order to do that is it increases your TPR and decreases your heart rate, which can be kind of complicated to think about in terms of our equation. Right, we had our heart rate, we had our stroke volume, we had TPR. So what I'm saying to you here is that we see, different color. Okay, so we're going to start with this heart rate part. So we're decreasing the heart rate. This is really what's kind of driving these effects. In order to increase CO2 elimination, one of the things that's producing a lot of CO2 is your heart. Your heart uses a lot of energy. It's very metabolically active. So if you decrease the heart rate and decrease the contraction in the heart, you don't produce as much CO2. So that's kind of what your body is doing. That's why it's slowing down the heart rate. But you can see a problem, right? If you slow down the heart rate, if that's the only thing we're doing, then we'd be dropping down our blood pressure, right? Which we know can be bad. That can be an urgent problem, right? We might fall, hit our head, depending how much we decrease MAP. So we don't want to do that overall. So what happens at the same time as your body is decreasing the heart rate is it increases TPR to kind of counterbalance this heart rate thing it's doing. So overall, there's generally actually an increase in MAP when you're trying to regulate blood CO2 levels, when there's too much CO2 in your blood, because your body really, really doesn't want to faint, even though it does want to get rid of CO2. So that's what's up with this. Okay. Also neat, thermoregulatory responses. So thermoregulation, right, is control of body temperature. This is really relevant to our lives like all the time. Thermoregulation actually takes precedence over the baroreceptor reflex, so over these blood pressure concerns, in terms of your body's hierarchy of like what variables about the body need to be under strict control. So your body cares more that you are the right temperature than that you have the exact right blood pressure. So thermoregulation is going to be controlled by the hypothalamus, and it's going to do a lot of different things to keep your body temperature the same. But the main way it can do this is by determining how much blood is getting to the skin, right? So what's happening at the skin, basically, right? You have your, you got skin, you got a bunch of capillaries here, right? So we'll draw some capillary beds. Beautiful, beautiful drawing. Okay. So what your body does if we want to change body temperature is it basically controls how much blood flows right by the skin here. Because what tends to happen at the skin, right, is a cooling effect. So as your body, as your blood comes into contact basically with the air, it cools down the blood. This happens for two reasons, right? So one, usually you're not sitting in a 98.6 degree room. But if you are, you're probably sweating, and the evaporation of sweat actually cools you down anyway, even if you're in a hot environment. So this usually works to decrease your body temperature. Okay. So this means that basically it decreases sympathetic activity to the skin. Do you remember constricted stuff? So it causes vasodilation in order to change your body temperature. So this is like a response to increases in body temperature rather than a reaction that does increase body temperature. I'll write that on here. So if we are responding to an increase in body temperature, we're going to decrease sympathetic activity to the skin, causing vasodilation, increasing heat loss to the environment. So that's all for regulating your body temperature. But if you suddenly have a bunch of blood rushing out to these capillaries under the skin, right? And especially if you're sweating, right? If you're basically losing fluid at the level of the skin, right? To create this cooling effect, which means you're losing blood volume, which means stroke volume is going down, right? Because there's not as much blood potentially, right? So this could basically decrease your blood pressure. So because this takes precedence over the baroreceptor reflex, this is decreasing your blood pressure. And basically, your body will kind of fight it out. You may see kind of cycling back and forth as the baroreceptor reflex and your thermoregulation kind of fight for control. Okay, so yeah, this last bullet point, this is true. So I was talking about sweating potentially decreasing stroke volume. Probably the bigger effect is the decreased total peripheral resistance from that vasodilation is also decreasing your blood pressure. So this is almost kind of a positive feedback loop in a bad way for your blood pressure as you're trying to regulate body temperature. So again, I'm just telling you this because I think it's neat and interesting, and I think that's why we're in this class, but if my explanation was too quick for you, don't worry about that for the exam. Okay, so this is just a graph of some of the things that change during exercise, because a lot of things are going on when you're exercising. So you can see how a lot of these have effects on your blood pressure, basically. So overall, we're increasing cardiac output because we're increasing the heart rate, we're increasing the stroke volume, but we may be dropping down the total peripheral resistance as we're dilating our blood vessels to get lots of blood flow to the muscles. We're also having effects on end diastolic volume. And basically what this is telling you is that the changes to cardiac output via changing heart rate and stroke volume are going to win out a little bit over any dilation that we're seeing, decreasing total peripheral resistance. So MAP overall is going to rise at least slightly, maybe more than slightly during exercise. So I'm just going to circle this middle. So you should expect your blood pressure to go up when you're exercising, which if you step back and take that kind of bird's eye view, hopefully, you know, you kind of had a gut feeling that that was probably true. Okay. We're going to do 2 like poll questions and then move into talking about red blood cells. I'm going to do these kind of quick because I don't want us to get too behind schedule, but I like talking about neat things about the body. So we had a bit more of an aside than I planned. Okay, so take another couple seconds. If you don't know the answer, I'll talk about it. Okay. So about half-ish of you chose this one. So I am gonna quick run through why this is. So what we're saying here is a person is standing up. Gravity can be pulling on the blood, pulling it to the floor, dropping blood pressure in most of the body. This would activate our baroreceptor reflex, which is those pressure receptors in the aorta and in the carotid sinus. So what we're looking for are changes that are part of the baroreceptor reflex, so that are part of the correction for this drop in blood pressure. So it is A, the decreased compliance of the veins. So dropping down their compliance, increasing venomotor tone, sort of tensing them, is one of the ways we increase stroke volume, right? Because we're looking basically for ways to bring our blood pressure back up. So we're trying to increase stroke volume, we're trying to increase heart rate, and we're trying to increase TPR. So A is the only one that would increase MAP. All of these other effects would decrease MAP. So we would actually see the opposite of these during the baroreceptor reflex. We'll see constriction of the arterioles, we'll see increased stroke volume, and we'll see increased sympathetic activity. And if you want to review this, this really is all about that flow chart. So this question is exactly which of these do you see in that flow chart. So if this was confusing to you, go back and take a look at that and spend some time practicing with that. Okay. This one I said we're not going to test on, so I'm not going to pull us directly on this, but this is asking us about that thermoreceptor stuff, which is basically that decreased vascular resistance to the skin would happen in this case. I'm not worried about it. Okay, so we're moving on to our cardiovascular system. Okay, so we want to understand each component of the blood. What I really want us to focus on mainly is red blood cells, because we talked, or hopefully you learned a bit about white blood cells in Phys Lab last semester. So we got a brief overview of composition of the blood, plasma, and hopefully today we'll do a chunk of talking about erythrocytes. And then we want to spend most of the rest of our time thinking about platelets and hemostasis. So we're going to try to talk a lot about clotting when we meet on Friday. Okay. But these are other topics that are in this chapter. Okay. So quick question. I think this might have directly been one of your Phys 1 questions. What's hematocrit? Awesome. Okay. Oh. Right. So the hematocrit is what our percent of erythrocytes, so our red blood cells, are in our volume of the blood. And second quick reminder, what's our largest component of the plasma? So we're going to take a little look at that test tube that I know we saw in Phys 1 in a second. Okay. So I think some of you got tripped up here by albumin. That's our protein that there's the most of, but plasma is mostly water. So plasma is mostly water. We've been talking about how water filters in and out of the capillaries and why it does that. So water is still the most important thing in the plasma. Okay, so I won't put this up as a poll, but this is where you should have put albumin. That's our most abundant plasma protein. And because that's our most abundant plasma protein, that's what's basically mostly creating our osmotic pressure when we're looking at filtration pressure in the capillaries. So here is where you would have chosen albumin. So we've seen this little blood tube before. So most of your blood, about 55%, is made of plasma. So that's water. We got some proteins in there. That's this yellow part. Okay. We got a buffy coat, so we got some leukocytes and platelets. So basically white blood cells, and platelets are going to help us with clotting. Very small in number, but have a very important role in blood. And then the rest of the blood is going to be made up mainly of erythrocytes, which are our red blood cells. Should be about 45% of whole blood volume, giving us a hematocrit of around 45. So we can see that there are different ranges for men and women, but 45 would be a normal number for pretty much anyone. Okay. Average blood volume is similar in men and women, 5 or 5.5 liters. About 3 liters of that is plasma. Right? The rest is mostly erythrocytes, but we got some leukocytes and some platelets. Okay, so we are going to spend a little time talking about the production of all of our blood cells and our platelets. And specifically, we're going to look in detail at how our red blood cells are forming. So this is a chart basically of where all the blood cells are coming from. So at the top, we can see we have our hematopoietic stem cell, which is a stem cell. So it's like a progenitor cell that can, depending on the right levels of the right hormones and stuff, turn into any one of these blood cells, right? That's why they're red blood cells and white blood cells, right? They are all blood cells. They are all coming from this original hematopoietic blood cell stem cell. Okay, so we can see that there are several different pathways that lead us to different places. So the hematopoietic stem cell splits into one of these other types of stem cells, either myeloid or lymphoid. Don't worry about those terms. And it's going to give rise to all our various types of cells. That is not the important part, particularly for today, but the term hematopoietic stem cell should be familiar to you from Phys. Right. So plasma is mostly water, like 90%. 6 to 8% is proteins. They're important, but they're not clogging up our arteries and veins, right? They're not big and chunky. They're there. The reason they're important is that they don't pass through our membranes, so they create that osmotic pressure for water to move across the membranes and into or out of the tissues. There are also electrolytes in plasma, roughly the same as in our other fluids. So we got high concentrations of sodium and chloride, relatively low concentrations of these other ions, hydrogen ions, bicarbonate ions, potassium ions, calcium ions. But you do got ions there. Okay. said I wanted us to talk about was the erythrocytes, which is true. So there are about 5 billion red blood cells per milliliter of blood, or sorry, 5 billion. It's a lot. We calculated out in Phys 1 how many red blood cells we could fit. So we know that the reason we care about red blood cells is really because we care about how much oxygen can get to the tissues. So the red blood cells are specialized for carrying oxygen, and we're going to see that all of their traits are really going to be linked to that purpose, including their shape. So they look like a kind of donut, except with no hole, right? We could refer to scientifically as a biconcave disc, right? Concave means curved inward, bi means two, so it's curved inward on both sides. It's a disc, it's like a frisbee looking thing. Reason this is important to us is that this creates a large surface area. So when we have a large surface area, right, we got a lot of membrane on the outside. This means that there's basically lots of room for diffusion into and out of the erythrocyte. So we're going to have lots of diffusion. They're specialized for diffusion. Oxygen isn't going to have to go very far anywhere in the erythrocyte in order to get to the hemoglobin, which we'll talk about, and it's not going to have to get very far to get out of the erythrocyte either. Okay. So they're pretty small, right? So their diameter is about 8 micrometers. Their thickness is about 2 micrometers. They are flexible, which allows them to kind of squeeze through capillaries. So one of the problems with sickle cell anemia is that the different shape actually creates some issues there. And that is an aside, but a neat thing. What you should know about what they look like, right, is that they have no nucleus or organelles. So in terms of cells, they look kind of weird, right? Like most of our eukaryotic cells, so most of our human body cells, they got a nucleus, they got all sorts of organelles, they got mitochondria because they need to produce energy. None of those things in our erythrocytes. They don't have mitochondria, they don't do anaerobic glycolysis. They get 2 ATP molecules maximum per glucose molecule. They are really pretty much nothing but like little bags of oxygen. Their major functions are to transport oxygen and also CO2. We will see that that's the case as well. Okay. So oxygen and carbon dioxide transport. So what we do have inside of our erythrocytes, our red blood cells, are this molecule hemoglobin. It's a protein. We'll see we also have this molecule carbonic anhydrase, which is an enzyme. So that's going to be our next slide. So the hemoglobin, its really important job is for carrying oxygen around the body. So the molecule overall, I just said it's a protein. It's made of 4 polypeptide chains. And inside each of those chains, we have what we call a heme group. So that's this kind of yellow flower inside each of these 4 little units, these subunits. We got 2 alpha subunits, 2 beta subunits. Okay? And inside that heme group, we have an iron atom. Okay? So this is going to be important. When we think about red blood cells, we're going to think a lot about iron. Okay. So the iron basically is what allows the hemoglobin to bind the oxygen. The oxygens are going to bind in at these heme groups. In your body overall, you can transport a little oxygen in the plasma, but like 1.5% of your oxygen is in the plasma. Not very much at all. Almost all of it, 98.5% of the oxygen circulating around your body is transported bound to hemoglobin, right? And when we get to the respiratory system, we're going to talk a lot about breathing and how those oxygen molecules bind in, when they bind in, when they have affinity for the hemoglobin and when they don't. But for now, we just want to understand that that is the role of hemoglobin, and they're in our erythrocytes. Hemoglobin with oxygen bound to it is bright red, and hemoglobin with oxygen not bound to it is dark red. So this is part of why if you see people bleeding, like it may look different colors if they've cut an artery versus if they've cut a vein. All right. Usually you don't want to see arterial blood because it's moving quickly, it's under high pressure. Arterial bleeds are not good. And practitioners can tell, right, when they see that bright red color, right, with that oxygenated hemoglobin, that they've gotten arterial blood because of that oxygenation. So like if you were to take a blood draw and instead of getting a vein, you got an artery, you'd actually be able to tell. Okay. Hemoglobin does also bind to some other things. So carbon dioxide, proton ions, and carbon monoxide. So carbon monoxide, you may have heard of carbon monoxide poisoning. It actually competes with oxygen to bind into those heme groups, which is why it's dangerous, because hemoglobin actually likes carbon monoxide even a little more than it likes oxygen. So it can block the binding of oxygen to hemoglobin at those heme groups, which is why you can die of it, right? You die of lacking oxygen, even though there is plenty of oxygen because your hemoglobin is all full. Okay. So we got the 4 subunits, we got a heme group. Important that the heme group contains iron, binds O2, carries most of the oxygen in our body. The other thing we have in our erythrocytes is carbonic anhydrase. So it's an enzyme. It's going to be really important for the transport of gases, which we will talk about as we talk about respiration. But basically what it does is it catalyzes a reaction where we take carbon dioxide and water and turn it into carbonic acid, which can then dissociate into hydrogen ions and bicarbonate. And both sides of this reaction are reversible. So carbonic anhydrase can turn CO2 and water into carbonic acid, but it can also turn back. So in the future, we'll be talking about like the pH balance of blood and how that relates to breathing. I think we talk about it in renal as well. So we'll talk more about these reactions in the future, but the carbonic anhydrase that we're going to need for those reactions is in our erythrocytes. Okay. So this is going to be critical to transporting carbon dioxide for one thing, right? We can bind it in. And it's also going to be critical to maintaining your blood at the appropriate acid-base balance, which we will talk about in the future. You don't want your blood too acidic or too basic. That would be very bad for your body. Okay. Yes, carbonic anhydrase is found in the erythrocytes. Okay. So if you're looking at a cell under a microscope, how might you tell that it was an erythrocyte? And you could actually like see this in a good enough microscope. Awesome. Okay, I'm going to unpulling. Right. You're not going to see a nucleus. You're not going to see mitochondria. You're not going to see a bunch of organelles that need to do things. Really, really bare bones kind of structure. Okay, and this one might be a little tricky, but what can hemoglobin bind to? See, answers coming in. Yeah. We focus a lot on it being able to bind into oxygen. It can also bind carbon dioxide. So carbonic anhydrous is going to be more important for carbon dioxide regulation, but it does also bind to the hemoglobin. So hemoglobin does both of these things. Okay. So I just said, erythrocytes have almost nothing in them. This means that they can't do mitosis, so they can't replicate, they can't really make copies of themselves because they don't have DNA, they don't have RNA, they don't have organelles, they don't have any of the machinery to divide, basically. They also have almost nothing in there to be able to fix themselves. They're going to have a really short lifespan in terms of cells. 120 days is the typical lifespan of an erythrocyte. And you make 2 to 3 million red blood cells per second. So on the order of 200 billion red blood cells per day. So we're constantly cycling through them. It's not like every couple months you replace all of them on one day. We are constantly making red blood cells. So they're synthesized in the red bone marrow via a process called erythropoiesis and filtered out by the spleen and also the liver. So they're going to be born in our red bone marrow, and then they're going to go die in the spleen. Okay. So here we're talking about their synthesis or their birth, right? So why, how are we making these red blood cells? So basically, your body knows to make red blood cells, and I should say here, really how many red blood cells to be making, because as we just saw, we're making them all the time. By stimulation through this cytokine called erythropoietin. So erythropoietin is a type of hematopoietic growth factor. Hematopoietic growth factor is basically this kind of thing that tells your stem cells which type of blood cell to form. So the one that tells them to become red blood cells is erythropoietin. It's secreted from the kidneys. So your kidneys are able to sense low oxygen levels in the blood. The kidneys are going to be responsible basically for filtering blood, so they can tell when the blood they're filtering doesn't have enough oxygen. If you don't have enough oxygen, the way you could fix that, right, is more hemoglobin, and hemoglobin is in our red blood cells, so we'd want more red blood cells if we're not carrying around enough oxygen. So when erythropoietin is secreted from the kidneys, this is going to tell those stem cells in the bone marrow to turn into erythrocytes, to turn into red blood cells. As the erythrocytes develop, they produce hemoglobin. And it's actually important here that we're highlighting developing, because in order to produce hemoglobin, a protein, right, in order to make proteins, right, like we definitely need ribosomes, right? We can't do it without some of those cellular components. So they produce hemoglobin before they lose their nuclei and organelles. So the hemoglobin is made first. You have a sort of normal looking cell, and then it loses the nucleus and organelles. It doesn't start out that way. By the time we call it an erythrocyte, it has lost those nuclei and organs, but like earlier steps in the process have their own different names while it's producing that hemoglobin. So this will be our last slide for today because we're at time, but so here what we're looking at, I'm sorry, I brought this image back from a previous slide, so I think it's probably not right here in your packets, but you should have it earlier and it's in your book. So we got our hematopoietic stem cell, right? So hematopoietic, it could turn into any blood cell it wants, but comes into contact with erythropoietin from the kidney, right? Your kidney says hematopoietic stem cell, I want some more red blood cells, I don't have enough oxygen. So it would start to divide. See, it's dividing just down this line. We're not going to name all these intermediate cells. We don't need their names. A name you might encounter in your future careers, though, are reticulocytes. So the reticulocyte you can see still has a nucleus. It's the step right before the erythrocyte. So sometimes under some conditions, like major blood loss or something, if you're producing red blood cells really, really quickly, you may still see these in the blood. So for some conditions, you'll actually do a count of how many of these developing erythrocytes you can see in the blood that aren't fully developed into mature erythrocytes. Okay. So during this kind of flow, right, we could be making our hemoglobin, but by the time we're a fully developed mature erythrocyte, we can't do it anymore. We have no nucleus, we have no genetic information, we have nothing to build anything with. So at that point, we'd stop making hemoglobin and we just circulate around, carrying our oxygen, working on carbon dioxide balance. Okay, so that's it for today, guys. Sorry that I spilled over a little bit. Take care.
Thank you.